I'm Christopher Johnson from NREL, and Paul and I today will be giving a brief presentation on some work that the ABF has done historically that can be leveraged toward potential industry partnerships. Next slide. One way that you could describe what the ABF does is that we develop synthetic biology infrastructure to reduce the time to market for biomanufacturing. So by infrastructure, I mean all of the sort of tools and things that can drive the design, build, test, learn cycle. And that's shown here on the right, where we've developed tools sort of along that whole cycle, including things like strain development, scale-up bioprocessing, machine learning tools, and all sorts of things to be able to develop strains and processes for production of various target molecules of interest to biomanufacturing. Next slide. And so when we think about how infrastructure enables private investment, you can think about it one way in terms of this this figure that was adapted from a Lyft campaign showing essentially how infrastructure, so in this case, a subway system, can enable people to get where they're going to a degree, right? So you may need something, you may be able to get close to where you're going, but ultimately to get to the door, you may need a Lyft or an Uber or a taxi. And so it's really that infrastructure investment that enables the private investment to get to where you want to go. Next slide. And with respect to the Agile BioFoundry, so we think of ourselves sort of as mostly that subway system in that analogy. So public investment in biomanufacturing infrastructure can enable private investment toward development, scaling, and tailoring of unique pathways and products of interest to industry. Next slide. And one way, sort of more concretely, that we have been working toward that is through the development of what we call beachhead molecules. So beachheads are metabolic intermediates that can be converted into several products. So the analogy here would be that a beachhead molecule would be a subway stop. And so if you look at one of the green dots on that subway map, that could be a stop that the Agile BioFoundry has developed tools and strains to be able to get to. And then by extension, we are usually tying that to a specific target molecule. So those are shown in blue. But as you can see on this map, you can imagine that if we've developed the infrastructure to get all the way to a certain subway stop and to one of the target molecules, that what we've done, that work can be leveraged toward other products as well. Next slide. So for instance, the metabolic engineering strategies that we've developed for a given target can be applied to targets associated with the same beachhead. And next slide. And the similar theoretical yields and processing parameters that are associated with the TA and LCA that have been developed for a specific target molecule could be extended to related products. Next slide. And so stepping out of that analogy and sort of into the real metabolic world, what's on the right is essentially a map of metabolism showing, and this is sort of metametabolism, this isn't any specific organism, but generally showing how you can get from, in this case, hydrolysate sugars, which is the primary feedstock that we work with in the Agile BioFoundry, to various products, which could be any of the things on that map. The beachheads that the ABF has been working with are shown in green, and the associated products are in blue. Next slide. And I just want to point out that the demonstration products or projects that you're going to be hearing about today are included as part of these. So Protocatechuic acid is the beachhead for muconic acid that can be converted to adipic acid. Pyruvate is the beachhead for ethanol that is one of the targets that we're pursuing for SAF production. And malonyl-CoA is the beachhead for 3-HP that's a precursor to many C3 chemicals, as well as alkanes that can be converted to SAF. Next slide. And historically, the Agile BioFoundry has made a lot of effort and progress in developing these beachhead molecules and the example targets that they're associated with. So there are so far 12 beachheads that we've developed strains associated with, and those are associated with 21 exemplar targets. We've onboarded 15 organisms. So these beachheads are sort of spread across those organisms depending on the various properties or characteristics of those organisms and how they're tailored for development of those beachheads or targets. And it's also worth pointing out that 9 of these products have been scaled to greater than 10 liters. And so this isn't, work that we've just been doing in microtiter plates and just evaluating strains in that context. This is more considerably, I think, more robust than that. Next slide. And this is showing that I guess in terms of numbers that might matter most to industry, this is showing a series of beachheads across the bottom that we've been developing and illustrating that a number of these we've actually demonstrated not only at considerable scales, but also achieved considerable titers. So as you can see here, there are several targets associated with beachheads that we have demonstrated at titers greater than 20 grams per liter. And I'm going to switch gears a little bit on the next slide and talk about protocatechuic acid as a beachhead and an associated exemplar target. And protocatechuic acid is there on the left. Next slide. Yeah, so like I said, this is protocatechuate or protocatechuic acid. The host here is Pseudomonas putida, which is a soil bacterium. And it has a robust ability to degrade and by extension in metabolic engineering terms, produce molecules related to aromatic molecule degradation. And so that's kind of was the, I guess, motivation here for using Pseudomonas putida. And the target that we've been pursuing is muconic acid, which again is derived from those aromatic degradation pathways. And muconic acid is of interest because it's a C6 diacid that can readily be converted to adipic acid. And you're probably more familiar with adipic acid as a high value chemical with a large market. That market is growing, and it's used for industrial applications like primarily as a precursor to nylon 6,6, but also in polyurethanes, plasticizers, and can be used in the production of PET as well. Adipic acid can also be converted to, or I'm sorry, muconic acid can also be converted to caprolactam and 1,6-hexamethyldiamine that can be used in nylon as well. And importantly, it can be used directly in the production of performance advantage bioproducts. And this is work that is being done outside of the Agile BioFoundry, but does motivate our interest in muconic acid. Next slide. And I'm going to take you through a little bit of a history of the progress that we've made with muconic acid, just as an example. So this is work, as you can see, published in 2019. So this was sort of in the early days of the Agile BioFoundry. And in this publication, we really um, set the groundwork for work that we've done since in producing this molecule. So essentially introducing the production pathway and deleting competing targets. And we made pretty good progress there in developing these strains for high yield production of muconic acid. So you can see there on the right, we got up to about 39% with a strain called CJ522. Next slide. From there, this was a paper a year or two later where we took that strain and performed adaptive laboratory evolution. And in this case, we were using a mucinate biosensor that was developed with an Agile BioFoundry at Los Alamos National Lab. And we're able to use that to pull out strains that grew faster, but also produced muconic acid more quickly. And you can see in the bottom right that CJ522 was pretty slow compared to GB271. So we were able to increase the productivity of the strain, and the yield stayed about the same. Next slide. So from there, GB271 was a pretty quick strain, but it only was able to utilize glucose. And so, as I mentioned earlier, hydrolysate is really the substrate that we're interested in. So with this publication and this study, we turn our sights towards xylose utilization, and along with input from Oak Ridge National Labs and work that they had done on xylose utilization and Pseudomonas putida, we were able to incorporate that. We did adaptive laboratory evolution and were able to improve the productivity using both glucose and xylose. So lots of metabolic changes here that ultimately led us to, as you can see on the right, a titer, in this case of 44 grams per liter, a productivity of almost a half a gram per liter per hour, and a yield of 43%. Next slide. And so most recently, this paper, I think, was just accepted for publication. We've been working on mostly bioprocess development for production of muconic acid using real hydrolysate. And so in this work, as you can see on the right, we compared real corn stover hydrolysate compared to mock hydrolysate. And that hydrolysate, of course, includes glucose, xylose, as well as a small bit of arabinose that in this study we weren't able to utilize, but that's work that's ongoing. And you can see that we achieved similar titers, rates, and yields with actual hydrolysate versus the mock hydrolysates on the left. Next slide. Something else that was also important that we demonstrated in this publication was the ability to scale up this process. So what you're seeing here is evaluation of these strains at a half a liter on the left, and then in a 150 liter bioreactor on the right. And we were happy to see, of course, that we achieved similar titers, rates, and yields in both formats. Next slide. And all of the strain and bioprocess development work that we do is really informed by modeling, and specifically technoeconomic analysis and life cycle assessment. What's shown here is the process configuration for production of muconic acid and conversion to adipic acid, everything from the seed train to production, muconic acid purification, hydrogenation to form adipic acid, and then finally the adipic acid purification. Next slide. And what comes out of that techno-economic analysis and life cycle analysis is shown on this slide and the next slide. So along the bottom there are the publications that I mentioned earlier, and where we were at with the techno-economic analysis and the minimum selling price of adipic acid and muconic acid. So as you can see here, muconic acid is a little bit cheaper in all cases because you don't have to, you don't have to convert it to adipic acid and have those additional costs. But what's important here is that we've been able to lower the minimum selling price where we're approaching cost parity with fossil-based adipic acid. So fossil-based adipic acid over the last several years has ranged from about $1 to about $2.50 per kilogram. The current adipic acid price, as I understand, is sort of on the high end of that right now. And so you can see with this most recent publication, with a little bit of addition of corn steep liquor up to between 1% and 5%, we're really able to get close to cost parity. Next slide. And this is just a peek at the LCA analysis. We've also made similar progress there, but you can see fossil-based adipic acid is really not favorable with respect to greenhouse gas emissions. So we've always been well below that threshold. And of course, that's one of the advantages of bio-based products and processes. Next slide, and I think that's where I'll turn it over to Paul.
Yeah, so my name is Paul Adamczyk. I'm staff scientist at Sandia National Lab. So yeah, I'll kind of discuss some additional work we've done with fungal hosts, specifically work on 3HP, for example, here. We originally started off producing 3HP with organisms such as Aspergillus pseudoterius and Aspergillus niger, as described here in a recent publication. And then later on, we tried to transition towards additional fungal hosts. So next slide. Here, we also demonstrated production of aconidic and malic acid and two additional organisms reaching titers of roughly 62 grams per liter and 25 grams per liter. Next slide. So effectively, main work I'm kind of talking about is related to an additional fungal host we have extensively worked with, which is the oleaginous yeast Rhodosporidium torreloides. Due to its natural ability to accumulate 70% of its dry weight as lipids, there is a significant acetyl-CoA flux, which we've historically leveraged, as noted by several beachheads stemming from the acetyl-CoA node on the right. Highlighted in the boxes and annotated with titers are all the target molecules demonstrated in Rhodosporidium so far. Of these, I'll be discussing the ones highlighted in blue. On the left are the beachhead target molecule pairings and their generalized utility. So for terpenes, for example, they can be saturated and used as fuels or fuel additives. Fatty alcohols as detergents, 3HP in biodegradable polymers, and sugar alcohols as sugar substitutes, and so on. Next slide. So as I briefly mentioned before, leveraging kind of the 3HP work and Niger and Pseudoterius in the ABF, we wanted to demonstrate our ability to make attractive bioproducts in multiple hosts and leverage previous findings to accelerate DBTL cycles to achieve comparable or better trimetrics in the transfer host. So for this specific example, there are three main ways to make 3HP. The beta alanine pathway was used in Aspergillus nigrogen pseudoterius. And for roto, we used the malonyl-CoA pathway. So the malonyl-CoA pathway uses the malonyl-CoA reductase, which catalyzes two steps to make 3HP. And previous studies suggested that splitting the enzymes in the enzyme into two steps improves titers, and our data on the left supports that. We scaled up to a 250 mil amber bioreactor to improve titers, but we quickly noticed that there was significant catabolism of 3HP. Next slide. To kind of address that, we utilized a high throughput functional genomics pipeline that we developed for Rhodosporidium and identified the gene responsible for 3HP catabolism. So kind of in short, summary of that method, a genome-wide barcoded library of knockouts, what's cultured on 3HP, and some control carbon sources. After several generations, you take a sample, you sequence them, and compare them to the reference sample to look for enrichment or dilution of certain mutant populations. And after that, you compile a list of knockouts that potentially are related to 3HP catabolism that you see in the middle here. And we identified after screening this gene ID 8975 was responsible for 3HP catabolism, which is a putative malonate semialdehyde dehydrogenase. This gene shares roughly 56% homology to the responsible gene for 3HP consumption and Aspergillus pseudoterius, further validating our findings and the utility of our high throughput functional genomics pipeline. Next. So lastly, for the 3HP part, we took the R torulades homolog to the transporter previously investigated in Aspergillus pseudoterius, and we found that it also improved titers in roto, demonstrating the portability of learnings from other hosts within the ABF. This transporter combined with the MCR overexpression, deletion of catabolism, led to the top strain you see on the right with the 2-liter scale up, achieving 45 grams per liter maximum titer. Next, please. So moving on to another beachhead downstream of acetyl-CoA is the pyrophosphate, farnesyl pyrophosphate, highlighted in the green circle, which is one step removed from alpha-bisabolene, a terpene that can be saturated and used as a diesel fuel additive. So on the left as well is the metabolic pathway of Rhodosporidium overlaid with the heterologous mevalonate pathway from Saccharomyces. So in our work, we overexpress the full 8 genes of the heterologous mevalonate pathway from Saccharomyces in a stepwise fashion, integrating the first 3 genes, followed by a subsequent addition of the last 5 genes onto a base strain that had multiple copies of the terpene synthase. So after a combinatorial stacking, titers approached roughly 13 grams per liter in the cornerstone hydrolysate in panel C on the bottom right. Another thing I want to note is that in panel A, under the large titer, improvement was due to codon optimization, was seen as pretty rudimentary, but subtle differences in methods used, although they had 97% sequence identity, led to big titer improvements. You can see in panel A from 6 to roughly 10 grams per liter. Moving forward. So taking a few of the top performing strains from the last slide, we compared growth on 300 grams per liter total sugar corn silver hydrolysate, supplemented with either of 2 nitrogen sources in batch cultures, achieving roughly 20 grams per liter bisabolene, which is the highest titer in the literature for any carbon source so far. On the right-hand side, next, we scaled up to 2-liter and 10-liter bioreactors and either of corn silver hydrolysate and a synthetic medium, achieving roughly the same titers in the top right panel of 19 grams per liter. And for one of the conditions in the 2-liter, we achieved a literature high productivity of 0.1 grams per liter per hour for this terpene. Next slide, please. So moving on, we explored xylose as another beachhead molecule in rhodospiridium toroides. So improving xylose assimilation rates and generally elucidating the fungal xylose catabolism was our kind of early focus. So leveraging multiomics data previously collected by our team, we found a putative regulator of xylose catabolism. Overexpression of this transcription factor showed a strong upregulation of L-arabinose and xylose catabolic genes, shown on the left in the big red arrows. In the top right, we then took this overexpression strain of the transcription factor and compared xylose consumption rates relative to wild type, and there was a massive increase in specific xylose uptake rate. Then we decided to apply this to a bioproduct, specifically fatty alcohols in the bottom right, grown on xylose, to show that overexpression of this transcription factor did improve fatty alcohol production. Next, please. So as a part of reconstructing our pentose, the pentose metabolism of rhodospiridium, we noticed that an unconventional catabolic route was being used that converts xylulose to D-ribitol instead of using the canonical D-xylulose to xylulose 5-phosphate via xylulose kinase. So as a result, the rhodospiridium naturally secretes the sugar alcohols, the ribitol, and xylitol. We then exploited this by knocking out single and multiple genes responsible for catabolism of these compounds, achieving in the top and the right, about top right and bottom right, 75 grams per liter ribitol and about a peak of 30 grams per liter xylitol. There are some issues with catabolism of xylitol due to redundancy of the pathway, which we're working on, but yeah. Next, please. So in addition to sugar alcohols, D-xylanate was another target molecule from the xylose beachhead. So xylanate itself has uses, but it is a few steps away from additional bioproducts, including 1,4-BDO. So our first step was to engineer rhodospiridium for xylanate production by mitigating competing xylose consumption for biomass production. So from a model reconstruction and multiomics data, we found that there are multiple xylose reductase candidates, and we've knocked out several, but only one that was able to mitigate consumption of xylose most significantly in the bottom right. Next, please. So after growth attenuation on xylose, we overexpressed 2 heterologous genes that catalyze the 2 steps to xylanate and tested growth on corn silver hydrolysate, achieving 86 grams per liter, which equates to 96% yield from xylose, with a peak productivity in the bottom right at 1.3 grams per liter per hour. Now, once we have this, since we have this high performance base strain, now it's possible to produce the additional targets that I mentioned in the previous slide. Next, please. So moving on to our current efforts, we are mainly focused around utilizing various feedstocks, particularly soybean meal, for production of precursors amenable to current SAF infrastructure. So since rhodospiridium is an oleaginous yeast, we're mainly focused on tags, so triacylglycerides, and fatty acids for processing with HEFA technology, for example. We have a 3-pronged approach outlined here on the left to achieve the target metrics of interest, including string engineering to funnel carbon towards lipids and decrease catabolism of them. Process development, so including feedstock choice and how it is pre-treated can really drive process economics substantially. And finally, to help us get there faster, we plan to leverage computational automation tools to get us there. Next, please. So to briefly summarize, our core message is target molecules from choice beachheads will enable us to rapidly develop the development similar target molecules due to high likelihood of shared engineering strategies and processes. Next slide. And finally, So, and finally, we only presented really a kind of subset of the details of the beachhead target pairings and hosts that ABS has successfully demonstrated so far. And we'll continue to expand on our repertoire over time to include such beachheads as those circled in gray on the right, for example. So with our established infrastructure and core message, yeah, we welcome any partnerships. And thank you.